think we'll give 1 minute for more people to join, and then we will start right at the top of the hour. Okay. Hello, everybody, and welcome to the PDA Mountain States chapter's first ever webinar. Our topic today is GMPs for cell and gene therapies. I am Suzanne Michaelo. I'm the Mountain States chapter president, and I'm going to kick things off with just a brief introduction to PDA, and then I'll introduce our moderators and our speaker, and we will dive into our topic. Oh, and my screen's frozen. 1 second. Okay, what is PDA? As most of you probably already know, PDA is a leader in providing information on science, technology, and regulatory topics to the pharmaceutical and biopharmaceutical industry. It's a great organization. It's a great place to meet people and to learn things. The Mountain States chapter of PDA was established to provide learning and growth opportunities for scientific and technical individuals in biotech, pharma, and medical devices. Our chapter strives to provide opportunities for members to participate in educational events, networking, and other opportunities. This is one of those opportunities, and we hope to have some in-person events once we get past the current restrictions. And we encourage industry professionals to join PDA, and we're open to suggestions to increase the knowledge and skills of our existing members. We want input from the members as well as dispersing information. This is a map that shows all of the various PDA chapters, so you can see we have worldwide reach, and becoming a member allows you access to information and input from all over the globe. We have many member benefits. We have a technical library where you can access any of the PDA technical reports for free. You can network with regulators and industry experts through PDA events and volunteer opportunities, including the PDA annual meeting, which is virtual this year and starts on Monday. So if you'd like to attend and you haven't registered yet, now's the time. You can read the latest research through the PDA journals. There's the PDA letter as well. There's local networking, professional resources like the online member directory and the career center, and then membership gives discounts on conferences, courses, and books. There are many different volunteer opportunities within PDA, including chapter officer, which is my personal favorite, and we are looking to bring more people into the chapter leadership for Mountain States, so feel free to contact me through the PDA website if you would like to volunteer. This is gives you kind of an overview of some of the tech reports that are available through PDA membership. There are new tech reports coming out every year, and members get 24/7 access to all of the ones that are in the portal. We also have PDA Connect, which is a good portal for sharing information between different members, and membership in PDA gets you full access to PDA Connect. There are a number of different member types depending on your status within the industry, so you can check the PDA website to see which one is most appropriate for your situation. And you can get rewarded for referring a colleague to PDA. So if you refer someone and they join, they get a reward, and you also get a reward. In addition, there is a great catalog of PDA books and publications, and membership gives you a discount on those books as well. Okay, for today's event, we're going to have two moderators. We have Sue Schneep, who is a longtime PDA volunteer. She's served in many different leadership roles within the organization, and we're very happy to have her involved with our chapter events. And then myself, Suzanne, I'm the president of the Mountain States chapter. And then we have our speaker, Dr. Kimberly Bytart-Hafen, who obtained a bachelor's degree in psychology at the State University of New York at Binghamton, and then went on to complete a master's and doctorate degree in neuroscience at CU Boulder. She completed 2 postdoctoral fellowships at the CU Health Sciences Center, where she specialized in embryonic and adult stem cell research. In 2005, she entered private industry with a position as a lead scientist at Navigant Biotechnologies. And in 2009, she accepted a position as a consumer safety officer at the FDA. Since 2006, she has been a principal consultant at Parexel, and she specializes in biologics, gene and cell therapies. She assists companies with the authoring of regulatory documents, interactions with regulatory agencies in GLP, GCP, and GMP regulatory compliance. Before I turn it over to Kim, I would like to point out that if you did not receive the handouts via email, they are available from the control panel. You'll see a little arrow with handouts next to it. You can download them from there. If you have questions during the presentation, you can type them into the chat, and we'll have them compiled and presented at the end of the presentation. And we also are going to open up the microphones at the end. So if you'd like to ask your question live, that is also an option. I will now turn it over to Kimberly to start her presentation.
I just wanted to start off by saying thank you everyone for your interest in attending this webinar. I do see some familiar names. Hello to you, and I'd like to catch up sometime. Thank you for attending. All right, let's begin with the unique GMPs for gene and cellular therapies. I'm just going to make sure I have control. Okay. All right, if we start with regenerative medicine, you can really see that there is a lot of clinical trials going on, and this is divided between gene therapy, gene-modified cell therapy, and tissue engineering. If you look at the largest numbers are in that gene therapy, gene-modified cell therapy, and that really goes back to the foundations in biologics, where you have a plasmid DNA with a vector used to transfect cells. And here we're going to either transfect them in the body with the gene therapy or transfect the cells outside of the body with that gene-modified cell therapy. And a perfect example of that is the CAR T. The areas that are currently being studied with gene and cell therapy, the largest one is oncology, followed by cardiovascular, central nervous system. As a neuroscientist, this is very exciting for me. If we skip over to the genetic disorders, this is very fascinating because they often are unmet medical needs. And here with gene therapies and cell therapies, we're able to give cures to these unmet medical needs from the past. Ophthalmology, this is very exciting because often the route of administration is difficult. And in ophthalmology, your eye is immunoprivileged, so you really have that excellent route of administration. And that's typically the biggest challenge for administering these types of products. So the trends in gene and cell therapy, we're seeing oncology and those genetic disorders, orphan drug products. They're really groundbreaking technologies, but they do address those small disorders, like where there's not a large population, but they are getting preference for approval. We have the potential for curing patients. And the regulations, they're behind the technology. So while the agencies are working to catch up, it really needs to be a collaboration with industry and regulatory agencies to help establish the how to manufacture safe and effective products. And that's really the beauty of this, that the agencies are willing to work with companies because these products are each unique. The clinical trials are unique. Now we're seeing more single arm, small patient, ethical considerations. How do we have the placebo, or do we have the placebo at all? And then we have these large treatment effects, which is very, very exciting. So it's high risk, high benefit, and then manufacturing. How are we going to manufacture in that GMP environment? If we take a look at this chart, what you can see is 20 years ago, when I was working in the laboratory, I was working with stem cells. You can see that there was a lot of applications for cell therapy. The gene therapy was lower. At that time, there was that issue with a death, with the bubble boy death with gene therapy, and it really stepped that technology back as far as fear of actually allowing that to go to commercial. And then 20 years later, now we've got the cell therapies commercial. Really was that breakthrough with the CAR T, opened the door for many cell therapies. Now we have so many clinical trials, which is so exciting, and gene therapy has proven to be safe and effective as well. So it really has taken that long, and I find it very exciting, and I'm so happy to be part of the industry helping companies bringing it to the patient. But these types of therapies are unique because it's fast. You start with your preclinical, and at that time, once you have the preclinical studies in place that you're showing efficacy and safety in the animal, you're going to submit that IND, but you can also submit for fast track designation, and then also that orphan drug designation. Once you get to the clinical data, early on, if you're seeing efficacy and safety, you can apply for the regenerative advanced therapy designation and the breakthrough designation. And this is going to accelerate your process through approval. So now you've got the clinical data, you've got the scientific data, and you're going to go fast track. So the difficulty is keeping up with the manufacturing. We'll take a little bit of a deeper dive into these designations. The fast track is if you are treating a serious condition and you have that non-clinical data. You could have clinical data, and you really want to be able to address an unmet medical need. With the fast track, you get frequent meetings with the agency and communications, accelerated approval, priority review, and a rolling review. So you can submit data as you receive it throughout your process. Breakthrough designation, it's the serious condition, but you're going to have that clinical data, and you want to show that it's substantial improvement over available therapy. With breakthrough, you get everything from the fast track, but you can also add guidance from the agency as far as how to plan your clinical trials, and you get a commitment from the agency. The regenerative medicine advanced therapy, it's a serious condition, you have that clinical evidence, and you have the potential to address an unmet medical need. With this one, you get the fast track, the breakthrough, but you'll get more interactions with the agency to help you, guide you through your process to your application, your BLA application. And the beauty of this is that you don't have to guess. They're willing to work with you. And the agency has excellent scientists that they're going to help you get this to the patients. Everyone has the same what they want. We want safe and effective products to the patients. So I just love how the agency is willing to work with the companies to accelerate the approval process. So what do you need for your CMC readiness for these expedited programs? Currently, there is approximately 100 breakthrough therapy and RMAT designations. most breakthrough therapy is gene therapy, and most RMAT is cell therapy. So CMC readiness is not a criteria, but it is very important to be on the pathway to show that you can manufacture this product. But what is really important is that clinical benefit for patients that have serious or life-threatening conditions. So most RMAT are awarded in early phase. Sometimes it's the same time as the IND submission. And the minimum requirement is to have that IND, active IND, but the minimal readiness for initiating IND may not be sufficient to get that expedited program. So it's really important to have that open communication with the agency to understand what their thinking is with your product and what's the best pathway to bringing it to the patient. The expedited programs are designed to accelerate clinical development. Typically, it shortens the time to approval by conducting pivotal trials. And if you are not ready with your CMC, it can delay your clinical program, and the FDA can choose to take back your expedited program designation if they do not see progression towards being ready to commercially manufacture. So here is the conundrum. We know the product works, large preclinical data, early clinical data. We know that you can help patients with unmet medical needs, but the manufacturing is not there. And a lot of these, specifically with cell therapies, they're so unique. So you really need to wrap them in the GMPs to be able to manufacture the same way every time. So this is the difficulty with these young companies that have the discovery to getting them to commercialization. So the quality standards for your early CMC for initiating the IND, safety comes first. We need to show that this is, it's not going to harm the patient. It's product specific, and it's risk and science-based approach. And it also relies on institutional knowledge of the product. So you can see, you really want to find that spot where the risk and the benefit align. The IND has a 30-day clock. First, there is that cursory review that to see if the IND is appropriate for submission and all the required sections have been included. A gap analysis is performed, and that's when you'll get that information request from the agency. And then there'll be a final assessment of to hold or allow to proceed. What is required in the CMC for an IND? You need to describe the product, the manufacturing process, the mechanism of action, analytical method qualification, auxiliary materials, they're human versus animal derived. We'll talk a little bit more about the importance of this later in the presentation. Master cell bank, master virus seed qualification, donor screening and testing. This is very important, and there's different unique obstacles, whether it's autologous or allogeneic. A lot of the allogeneic, there's some nice companies coming out being able to produce GMP cells, which is really ideal. Autologous is a little bit different, where you're going to rely a little bit more on the clinical setting. In-process release specifications, drug substance release specifications, drug product release specifications. This is very important. Sterility, identity, purity, viability, mycoplasma if it's cultured, and potency is very important. And then any other ones that are product specific. You need an action plan for sterility failure when results are not available prior to administration. Sometimes these tests take time and the results are not available till after the product's been administered, so you need a contingency plan. You need to know container label. That's very important with the agency. The product needs to be appropriately labeled and the proper instructions for use. You need stability studies to help support your shelf life. Shipping qualifications, so important with the cell therapies as far as the chain of custody and keeping the cells viable. Sometimes they're going across the world to the manufacturing site and back to the patient. And then environmental impacts, and this one is interesting. The agency's been asking for shedding studies recently, and you look at saliva, urine, all feces, all kinds of excretion to make sure that the product is broken down properly and is not being emitted into the environment. It's a life cycle approach. So you're going to have your GMPs, your manufacturing controls that include, most importantly, prevention of contamination and cross-contamination. A lot of companies are using CMOs and they're manufacturing many different products. So this is very important. Manufacturing consistency and product quality. So it's a spectrum as you move from first in human to licensure for GMP compliance, and you really need to ramp up, but by the time you get to your pivotal, you really want to be at a GMP level because this is accelerated, and as it follows the pathway of small molecule, it's faster, so faster to that GMP. So here we have a picture of my love neurons, very beautiful for that central nervous system, and also a cell therapy. So what is critical for CMC readiness? For allowing the IND to proceed, you need to focus on safety. Yes, if the product works, but you're gonna hurt a patient, it's not appropriate. So that's most important for the expedited program designation. And the pivotal studies cannot be initiated without sufficient phase-based appropriate product manufacturing control. So CMC readiness for initiating the IND may not be sufficient for receiving the expedited program designation. It really is that collaboration with your clinical, your safety, and your readiness to manufacture. It's just the perfect combination of what you need to show to the agency. So the FDA considers and may accept clinical data from a comparable product, but the assumption is that the product manufactured by the sponsor is manufactured comparably to the product that is referenced. So to show that you're manufacturing in a state of control, oh, and here we have a picture of a T cell, it's beautiful. It should meet the following criteria, and this is where we get into the quality by design. You need to know your critical quality attributes and your critical process parameters and your key process parameters. And this is how you show that you're manufacturing consistently at the pilot scale and that the pilot scale represents the commercial scale. You need to show qualification of the process, and your release specifications need to reflect product quality based on the empirical evidence that you have at that time. Your assays should be qualified or validated, and that goes along a spectrum to commercial manufacturing. Supply chain is so important because with a lot of the cell therapies, you're not able to do that 0.2 micron filtration or a lot of the viral inactivation steps. So it's important that you know what you're putting in your product. And then you need to plan for a manufacturing process change and manufacturing site change. It's common for companies to do their clinical manufacturing at a contract manufacturer and then switch to commercial at another contract manufacturer. So that's going to be tech transfer, and the one for commercial is going to be more of that larger scale. So that you need to be prepared for that if you're going to make that change or if you're going to invest in the manufacturer initially for both sides of manufacturing. So knowledge of the product, so important. Mechanism of action, the target product profile, critical quality attributes, critical process parameters, and a risk assessment to tie them all together. So the risk assessment is going to link your critical quality attributes with your critical process parameters. It's not a substitute for lack of product knowledge. It's science-based, it's performed early, but it's part of the life cycle. So you always revisit. You have your risk assessments or living documents, and you always revisit them. It's a tool, and more information is in the ICH Q9. And for expedited programs, you really have to have the knowledge of those CQAs, CPPs, and their relationship to product quality. If we take a look here at the risk assessment, and it's the life cycle approach. So from development to process characterization, to process performance, to continuous improvement, you're gonna look at the severity of effects of events associated with your CPPs on critical quality attributes, your KPPs on process performance. You're going to look at the occurrence probability of the event, the detectability of the event, and then you can rate it based on risk. So you mitigate the highest risk and you monitor the lowest risk. A little bit more into the quality by design. You're going to establish that quality target product profile. You're going to use to determine the critical quality attributes. For the risk assessment, you're going to link the process parameters and the critical material attributes to the CQAs to define the CPPs and the KPPs. And then you're going to define a design space. And the agency really likes this, and it is to your benefit. And you really want to, in your process validation, test your design space. And this allows you to have flexibility in your manufacturing while showing that you still produce a quality product. The design and control strategy using risk management is accomplished by linking the CQAs to the process capability and detectability. And this is always going on with a continual improvement for the product lifecycle. You want to monitor these performance parameters over time to show that you are manufacturing consistently. Another important part of the CMC section is the analytical method development. So you want to, for your analytical methods, define specifications with the upper and lower limit. You use historical data and inherent assay variability to define these boundaries. Throughout your clinical manufacturing for the trials, you're going to learn more about the process and assay variabilities, and that will help you to further develop and validate these analytical methods. Your specifications for your product should be reflected of your CQAs the identity, purity, and potency. So with your spectrum of going to GMPs, you're also going to have this spectrum of analytical assay development. First, you start with the qualification to show that it's suitable to measure the analyte. It's sensitive, accurate, reproducible. It's required for the early phases of clinical trials, and it's for all of those critical analytical methods. If you use compendial, then you want to follow those sampling requirements that are listed. As you move towards your pivotal trial, then you want to move towards validation of your analytical assays. And here you're going to show that they're suitable for their intended use under worst case condition. So they're sensitive, accurate, precise, robust, rugged. Sometimes you'll look at different labs testing and by different operators, and you're really showing that this assay is suitable for its intended use and reliable. My favorite assay is the potency. It's the most difficult one. Here you're going to show that your mechanism of action is working for each lot manufacturer. You can do it in vitro or in vivo. In vitro, using perhaps a cell-based, is going to be the most reliable and cost-effective. In vivo, you have variability with animals, and you really should move away from that, just so that you can have a accurate method for potency, because you're going to be testing it a lot. Stability, lot release, your DSP. So I recommend in vitro. I love the scientists that create these assays. If I went back to the lab, this is what I would do. You want to have this qualified early around the IND, and then you want to have it validated when you are going to your pivotal. But it definitely is a challenge. Supply chain, very important for your CMC, raw materials, auxiliary materials, equipment containers. So because some of these therapies, you're not able to filter or activate different kinds of organisms, you want to know about your source material collection equipment. So here could be, for example, apheresis equipment, media and growth factors, manufacturing tools, flasks, bags. I hear from companies that have a difficult time finding GMP grade, but also you can't trust these companies. They'll tell you it's GMP, but it's your responsibility to qualify your vendor and go in there and make sure it truly is manufactured GMP. Container closures, bags and files, QC test platforms. So shipping and storage are important, and chain of custody, especially with the cell therapies. If you're having an autologous product, the chain of custody, you cannot transfuse it into the wrong patient. So very, very important. Material qualification, identity, safety, purity, potency. You can't rely on a certificate of analysis. You want to make sure that you're testing your products for identity at minimum when receiving them. Vendor qualification is so important for your materials, especially your critical materials. You want to make sure, you want to verify that they have that good manufacturing practice. Quality agreements outlining who's responsible for what is very important. They're not going to hold up in the court of law because you are the sponsor, you are responsible, but it's something that you can have between yourself and the CMO up front to define who's responsible for what. And it's so important to have a good relationship with your CMO so that you can trust what they're doing, and also obviously with your vendor so that you can trust the product that you get. Alternative sources, very important, because if you cannot get a raw material, you cannot manufacture. So you really want to have those alternative sources for your raw materials and components. Manufacturing changes, it's inevitable as you scale up and scale out. Automation, so important. It's just a really nice way to be able to follow those critical process parameters. Change of reagents, manufacturing site change, that does sometimes happen between through the life cycle of your product. And the sponsors are responsible for that comparability to show that any changes to manufacturing did not impact product quality. And you want to demonstrate that the product is similar to before and after the change. And more information is outlined in ICH Q5E. And you can show comparability with in vitro or non-clinical, but it's not always sufficient, and you may be asked to perform additional clinical studies. So for the comparability, if you have good knowledge of your CQA and CPPs, and that your CQAs are correlated with product quality, safety, and efficacy, you could demonstrate that before and after the CQAs are the same, and then they comply to a predetermined acceptance criteria. But if you don't have this good knowledge of these CQAs and CPPs, you can look at the product attributes before and after the change, but you're really going to want to step it back also to look at the in-process testing to show that they are comparable. And this may not be sufficient as well, so you may need to do more work beyond that. You want to look at the manufacturing yields, your key process parameters, the risk assessment, so important throughout the life cycle of the product, and then process qualification, validation after any major change to manufacturing. So the more reflective your CQAs are of clinical outcome, the easier it is to show comparability. What's essential for comparability is a description of the proposed change, the risk assessment, a rationale for the proposed changes, comparability study designs, ranking of critical attributes for product safety and activity, comparability assessment of quality attributes before and after change, side-by-side comparison using the same biological source material as preferred, and a justification for the acceptance criteria for establishing analytical comparability, and detailed analytical procedures, sampling plan, statistical method, and analysis. And this is a picture of CAR T, the T cells attacking the B cell. So CMC requirements for licensure, if we move a little bit farther down, when you're considering the BLA application, you're going to really want to get to that GMP level of manufacturing, and this will be verified at the time when you are given licensure approval. But you really want to plan early and continue on that spectrum after you get commercial licensure. So some issues that occur with the BLA review, if you haven't adequately validated your process or have a good plan for it, if you don't have sufficient stability data, lack of proper validation for analytical procedures, insufficient information about extractables and leachables. And this is really important because the platform is really moving to more of a disposable system. And that's how a lot of CMOs can manufacture different products while reducing cleaning validations, and just, being able to manufacture those different products. So here it's important the whole way through the process, nothing is going to impact your product. adequate SOPs, lack of retention samples, lack of shipping validation, inadequate donor eligibility determination, and that's really something unique to the cell therapies. Insufficient information about CQA, CPP, lack of container closure integrity tests, lack of validation of the starting biological material, lack of information about formulation, lack of validation of dilution or thaw wash step at the clinical site, also unique, where you're going to have those instructions for use so the clinicians are able to administer your product, and deficient content on the label. The other component, you're going to have your BLA review, and then you're going to have your inspection. Issues that occur in the inspection include inadequate quality systems, incomplete process validation or plan for validation, and validation of aseptic processing. So important because you're going to rely on this and your raw materials in cases where you can't do the additional inactivation or filtering to assure that your product is safe. Batch record documentation, lack of detailed process description, inadequate material qualification. You want to make sure that you're looking at the identity tests for incoming materials. Inadequate contamination and cross-contamination controls, so important, especially at a site that's manufacturing multiple products. Also, you want to look at the flow when you're auditing a CMO to make sure that there's no potential for material and personnel flow for cross-contamination there. The segregation and quarantine of your raw materials, also of your products, the DSDP, they need to be released before they can be used. Inadequate SOP, change controls, investigations. Something that I see a lot with these young companies is trying to start out in a paper-based system. This will work in your clinical, but if you're going to manufacture large scale, you really need to move to electronic systems where you'll be able to track and trend your quality indicating parameters to show that you have control over your quality systems. Inadequate chain of custody, it's so important to know, especially with autologous, where the cells are, that they're going to the right patient. And we need to know the maximum manufacturing capability. So when you're preparing for licensure, you want to have a validation of the collection materials. So you really want to know, especially for cell therapy, that they're tracking the appropriate information for the collection materials. And this is a challenge in the clinical setting because while they have been collecting these types of cells, for example, apheresis for years, they are not used to the documentation associated with GMP, and this really needs to be something that is a change to the clinical environment, because if there is an issue with the product, you're going to want to go back to that initial starting material, and you need to have the documentation of how it was collected. So this is a current challenge. I know with some of the apheresis, they rely on technicians to download the data when there's a problem, but you really need to know what was done during the collection for every single batch. Extractables and leachables, so important. You need to know if they're going to impact your product at any point of the manufacturing. A challenge is measuring for particles, especially with these cell therapies. You want to perform a risk assessment for any potential sources of particulates. You want to reduce that introduction in the manufacturing environment and in your components. And you want to develop tests for measuring and monitoring visible and sub-visible particles. And what you can see, a theme from this, is that gene therapy is a little bit easier to manufacture, than cell therapy because it really follows the manufacturing of different kinds of the gene therapy has been going on for a long time as far as the manufacturing, and the cell therapy is just so unique in depending upon what you're trying to do. The gene therapy follows a little bit more on how you manufacture a vaccination, which we have a lot of history with. The final drug product needs to be in a single final container. You need to understand the container quality, sterility, pyrogenicity, particulates, leachables and extractables. Fill finish needs to be in an ISO 5 environment. You want that aseptic process validation. You want your containers to be transparent so you can perform that visual inspection. And it's ideal to be able to sample from the final container. If we look at process qualification, and the picture here is of gene therapy, you have your upstream and your downstream and your fill finish. For process qualification, you really want to make sure that you lock down those manufacturing changes, which will just reduce how many times you have to qualify your process. Process qualification is going to show that you're manufacturing consistently to predetermined specifications that are reflective of product quality. You can perform it based on critical unit operations, so you could qualify the upstream, downstream, and then fill finish separately. And you could use your pilot engineering runs as the qualification if they represent the final process. Aseptic process validation is so important. Media fill can help you assess if you are not introducing any kind of components to the product. Most products, except for viral vector plasmid genes, cannot be sterile filtered, so it's so important that you're able to manufacture in an aseptic environment and prevent the contamination from the environment or the operators. So considerations for regulatory and compliance. This is where, it's really hard in this field. You're moving from academic to CGMP fast. You're skipping some of those phases. So you need to get to that commercial scale by the time you're at the pivotal studies. So virtual companies, so common these days, they really need to perform that due diligence to decide if they want to manufacture in-house versus CMO. They need to know their criteria for selecting the CMO. They need to understand the selection of the facilities for the starting materials, selection of the CMO or procurement facility as far as collecting the cells. You need to know about quality oversight, material supply management, donor eligibility, good tissue practice, good manufacturing practice, and this is the responsibility of the sponsor because they own the product. Tech transfer is important, especially if you were going to start with your initial technology from discovery or if you were going to move from one facility for manufacturing to another. Facility audit to make sure that they have the right controls and compliance. And you want this good relationship with your CMO so that when the agency goes to their site to check how they manufacture your product, there's not going to be a problem. So a lot of companies will work with their CMO and the sponsor to make sure that they're ready for inspection. In conclusion, you really want to get to that. It's a spectrum, but you want to get to that GMP compliance by the time you're ready for your pivotal trial. But the agency is going to check this when they are looking at your licensure. You want to plan for this before licensure and continue thereafter. You want to consider this for your CMC of your BLA. And you also want to consider this for when you have your licensure inspection. Thank you. And I also wanted to thank Mo Hydran. He's also a colleague of mine at Parcel who contributed to this presentation.
All right, thank you very much, Kim. We appreciate your presentation, and I think we are ready to take some questions.
We are. That was great, Kim. It was lovely. I really learned a lot. I thought and this is the first one we've done, so I thought it went really well. And I did not get any questions coming in over the webinar, but I did have 3 that were submitted prior to the presentation. So I think we should jump right in with those. And we'll see if then anybody wants to ask the question live. But the first question that we have is, what analytical method should be used to perform genotype and phenotype? Should we use NGS sequencing and karyotyping, or can we perform one of them? So it looks like they want to know if they have to use both or they can just use one.
Okay. So your PCR-based analysis are the best for this. NGS, next generation sequencing, is really the cutting edge, and you can perform your genotype and phenotype using this type of analysis.
Okay. Do we have anybody on the audio who wants to ask a question? Any brave soul? You're so quiet. So let's jump into question 2, which is scaffold. Is that drug, medicine, or medical devices? Maybe it would help to explain a little bit what that scaffold is.
So the scaffold it's really exciting technology, and we see that more in the cell-based regenerative medicine, where you can use a polymer scaffold to seed your cells, and then you transplant it into the patient, and the cells grow on the scaffold while the scaffold degrades, which, say for example, cartilage. So over time, the scaffold degrades, the cells grow on there, and then you're left with the cells in the body where you need them. So that's what I think of with the scaffold, and when it's used with the cells, then you have a combination product. The scaffold alone would be a device.
Okay. We did have a question come in on the question line. With the COVID-19 situation now, do you have any experience with FDA performing virtual audits for PAIs?
Okay, they did start with virtual audits, but they just come out with saying that they're going to go domestically on site. It's going to be announced, which is really nice because of COVID. So they want to make sure that their investigators are safe. So they'll discuss how their investigators will be safe while on site. So that is opening up. But my foreign clients are having the remote. And at Parcel, we've been performing remote. And then we're starting to go on site in August.
Okay. For the foreign clients?
Well, I guess it depends if we open up or not. But yeah, because and that would be the government guidelines. If it opens up, everyone will go. But the industry has changed where these remote audits are becoming effective. I personally used a virtual tour where I can interact with the client, and it's been very efficient, and you can do that, review documents. So it's kind of a blend, but I think if you are domestic and you have a PAI with the FDA, it's going to be on-site announced. But things could change if we have the second wave, right?
Absolutely. Here's someone who's new to gene therapy and they want to know if the drug product is considered as a drug device combo product.
For gene therapy?
That's what it says. At least that's how I'm interpreting it. It says new to GT, gene therapy, is DP drug product considered as drug device combo product.
Okay, it's not going to be a device, it's going to be a drug.
Okay. This is an interesting question that came in prior to the webinar. As the requirement to perform viral inactivation on manufacturing of biological product, what method is used to perform viral inactivation on processing cells in gene therapy? And then there was an example. For example, in mesenchymal stem cell, MSC, size of the cells are higher than sterile filtration using 0.22 micron. So this method is not acceptable for this product. What is the proper method and how should we validate that inactivation method? I'd throw in there too. Do you think there's going to be a requirement for people to go back and do viral clearance on COVID-19?
Yeah, I see what you're saying. The problem there, like for gene therapy, it's easier because you can filter it. Biologics, the product is a protein, you can filter it. But the cell therapies, as far as particulates and the viral inactivation, you have to go back to your materials that you're using and see how they're qualified and tested. And then you also need to have that aseptic environment and then test your DSDP. But as far as the actual act of inactivating, it's not possible. So it's about the controls.
Okay. Here's another question we had. What method of visual inspection for a drug product, cells in a bag, should be used, considering variability of the starting material, human apheresis cells?
Yeah, it's a challenge. Yeah, you gotta do the visual. Can also sample and do the classical visible, non-visible particles, but also knowing that the cells are going to be in there. But it's definitely a challenge that's unique to the cell therapy because you're also looking at the cells, and that's once again you have to go back to that. The source material, environmental controls.
And the last question we had submitted online was what common controls should be implemented to account for product variability in the case of autologous donors? Sorry about that. Autologous products tend to have high variability leading to possible process changes throughout development.
Yep, it's true. It's true. When you get to the allogeneic, it's nice because they really can lock down those GMPs, but the autologous will always have that variability because each product is one unit. It's small, and you also have that additional layer of the chain of custody. So often companies with CAR T, they'll require some of the testing for virus or infections to make sure that they can at least have a starting material that's not contaminated with some kind of virus or bacteria, and that can increase the consistency of the manufacturing. But that is pretty much the controls that you can put into place.
Okay. And the last question I have is, and I think it's the last question we have time for, since CT involves combo products, do we need to comply with Part 4?
Okay, so combo products, you're going to have to comply with device and pharmaceuticals. So it's an interesting challenge, being able to have that whole design control and also your classical GMPs or the pharmaceutical. But really, if you use that quality by design model, you're able to comply to both sides simultaneously.
Okay. And that usually, if it's a combo product, does the FDA not have a combination products like group that kind of handles it and does some of the liaising with the different divisions in FDA to handle those?
Yes, and then this is also a unique area because now we're involving not just your classical small molecule, but we're going to have these unique cellular therapy technologies. So they would also work with the Office of Tissue and Engineered Products as well. So it's unique. The agency's evolving. They're evolving with the industry and with these unique products, and it's exciting.
I have just one last question and then no more, I promise, because I know we're running over. In Canada, do you know whether the preclinical studies for CAR T studies have to be performed under GLP?
So preclinical studies in Canada under GLP, well, you'd really want to have that. You can do your analytical under GLP, but you really want to have that GCP for your clinical. And then at the same time, and quality is quality, no matter where you are in the world, you want to have the controls in place to show that you're consistently manufacturing, on that spectrum to the point where you're going to commercialization.
Okay. That really is all we have time for. So I'll turn it over to you, Suzanne, to close us out.
So I would just like to thank Kim for doing this presentation for us. I'd also like to thank Sue for moderating the questions, and a big thank you to Shanna Morgan, who's the manager of membership and chapters at PDA National. This would not have come together without the help of all three of those ladies. And thank you to all of our attendees, and I hope to see you in a future Mountain States PDA chapter webinar. Thanks so much.
Thanks, everybody.
Thank you.
Are we done recording? I'm afraid to speak.
I think it's still recording.